Good afternoon and welcome to Comet Corner. This is a virtual series created by the UT Dallas Office of Research and Innovation in partnership with the schools across our campus to host chats with some of our notable alumni to hear how they have taken their research out into the world and made an impact. My name is Enrique Ziller, Research Education Programs and Outreach Manager in the Office of Research and Innovation at the University of Texas at Dallas. As we move through this session, I invite you to become part of the conversation by posting questions and comments into the chat for our guest speakers to discuss. Here to guide the conversation and introduce our guest speakers is today's moderator, Giuseppe Miranda. Giuseppe is a doctoral student studying cognition and neuroscience at the Center for Vital Longevity at UT Dallas. He is already an alum of UTD, having obtained his master's degree in applied cognition and neuroscience in 2019. Giuseppe earned his undergraduate degree in psychology at the University of Dayton. He is interested in brain structure, aging, dopamine, cognition, and the impact of health and lifestyle factors have on healthy aging. Giuseppe is also fascinated by data analysis and statistics, both in his research lab work and a personal interest in finance. In his free time, he enjoys playing basketball, exercising, working on art projects, and spending time with friends and family. Giuseppe, welcome to Comet Corner.
Thank you, Enrica. It's really a pleasure to be here, and I'm very excited about the work that we'll be presenting for you all today. We have two great talks planned from two very interesting lines of research with two unique perspectives. The content is going to range from how a normal population ages to how those with psychosis age. And I really feel that with these presented together, as you'll see today, that we will all be able to gain a perspective that really wouldn't be as cohesive if we were just looking at aging from one of these perspectives. And I am also hopeful that we can have a great discussion at the end of both talks today as we learn more about the role of aging. So yeah, everybody, please keep the questions flowing throughout the talk and we'll address as many as we can at the end. And with that, I have the pleasure of introducing our first speaker, who happens to be my PhD advisor, Dr. Kristen Kennedy. Dr. Kennedy is an associate professor of psychology in the School of Behavioral and Brain Sciences and the head of cognitive neuroscience at the University of Texas at Dallas. Her research lab is housed at the UT Dallas Center for Vital Longevity. And she's also the Associate Professor of Advanced Imaging Research at UT Southwestern Medical Center and an affiliate of the Alzheimer's Disease Center there. Dr. Kennedy's research on the cognitive neuroscience of aging centers around the health and lifestyle modifiers of brain aging and their cognitive consequences using multimodal imaging techniques. This research has continuously been funded by the National Institute of Aging, beginning with an award of a K99 and an R00 grant in 2010. Dr. Kennedy has published more than 75 papers on cognitive aging and brain aging in the premier journals such as Cerebral Cortex, Neuroimage, and Journal of Neuroscience. She also currently serves as the senior editor at Neuroimage. So Dr. Kennedy, please tell us more about your research.
Hi, thank you to the organizers for allowing us to come and kind of put our research together in this unique way. And I hope that you will take a lot of new knowledge away from this today and that you feel very welcome to participate in the discussion. We're happy to answer your questions if we can. And so of the aging and psychosis piece, today I'm going to focus on what is normal aging. And this may seem as if it's a bit of a cheeky question. How can we not know what normal aging is? But it's actually quite surprising that we don't completely understand normal aging yet. And so even to the untrained eye, if I ask any of you in the audience to look at these brains, I think that any one of you, neuroscientist or not, would be able to come to the conclusion that, let me get a pointer here for you. I think any of you would be able to come to the conclusion that in this kind of autopsy cartoon of a brain, that this would be the individual who is aging more, quote, normally, and this would be the individual who is aging less normally. And so, of course, you would be correct. And so this is the person with what we would deem a healthy brain. This is the person's brain at the stage of advanced Alzheimer's disease. And so one could be tempted to stop and say, what is normal aging? Well, maybe normal aging is just the absence of disease. But I hope to convince you today that normal aging really is far more complicated than just the absence of disease. And this is kind of a recursive inverse interpretation that's not very satisfying. And so we can do a few steps better than this. One thing we can do is peer into the brains of individuals while they're still alive. We use that mostly using MRI techniques. And so again, you don't need to be a neuroscientist, I don't think, to be able to identify that this MRI of this the sagittal slice to the middle of the brain is from a younger, healthy individual. And this brain over here, this image, is from an older individual. What you wouldn't know just from looking at this, but that I know because these are participants from our research study, is that this is a very healthily aging older adult who is very much cognitively normal. And I can tell all of you who haven't hit the age of 80 yet, who are in the audience, you very much hope that this is your brain when you're 80. This is a very, very well-off person. So where do we stand then on this topic of what is normal aging? And so I'm going to continue to try to convince you that normal aging is more than just looking similar to when we looked when we were young, or looking more similar to younger adults. And so to do this, it's helpful to broaden out and think about the person kind of in the whole context. So when you think about normal aging, you also want to think about cognitive aging. In the end, if we are very cognitively healthy at the end of our lifespan, do we care what's under the hood? Not so much, right? So if my brain looks awful when I'm old, but I'm perfectly fine, I'm happy with that too, right? So probably the more important metric is our cognitive performance as we age. And so here are two instances of domains of cognition that tend to not fare so well with the process of aging. And so here are, each dot is an individual subject in the research study, and then it's plotted on the x-axis along age. And so this, for example, would be an 82-year-old subject. And then up here, we have the scores on their cognitive tests, with higher scores being better. And so as you can see, the 20-year-olds perform up here. And then by the time we're in our 90s, we're performing more poorly. And you can see that this is a significant declining slope with age, as you would expect. Here's another example, same thing, it's just executive functions instead of working memory. And again, you can see an even steeper slope here. And so the real question, the interesting question, is what is leading to and building up all of this heterogeneity in cognitive aging that we see across individuals. And so you can see, for example, here are older individuals who are scoring quite high, on par for certain with middle-aged adults and some younger adults. You can see this in both domains. You can also see that there's individuals who aren't performing as well, and they're down here in this bottom quadrant of the graph. And so really the biggest question, I think, in our field of research, what most of us study at the Center for Vital Longevity, is what determines how an individual is on track to end up here or here, and can we intervene at any point to try to bring people up to a better aging trajectory? That's basically the whole point of our field of research. And so then you can ask, well, then is normal aging just staying above the mean regression line? Maybe, but knowing all the things that are going into what makes us land there, I think is probably the most interesting part. And so one of the ways that we do that is we put together all of the pieces that we can easily study in vivo in living individuals, preferably repeatedly over time to see how each individual is aging. And so here's kind of our conceptual model of this process that we kind of work from. And so this is aging of the brain and its effects on our cognitive output. And you can see that this is a complicated mixture of multiple synergistic influences. And we could come and give a talk on any one of these boxes, basically. Today, what I'm going to try to convince you is that age is just kind of an umbrella term that literally is just tracking how long you've been alive on this planet, right? It's much more interesting and important to know what your genetic predispositions are, what health and lifestyle factors have influenced you throughout your lifespan. These all have an effect on your brain structure. Your brain structure, ostensibly, is what allows your brain function to arise. And then some mixture of this, which is what I'm exclusively going to talk about today, is then what kind of sets us on a trajectory toward Alzheimer's disease or other types of pathology, or on a healthy aging trajectory. And again, in the end, I think what we all care about is preserved cognitive ability. And so that's always our outcome measure that we keep at top of mind as we think about these things. So the goal of understanding this complicated mix of synergistic influences is for ultimate clinical guidance for better, more healthy cognitive aging. So how do we study this? If we're going to study something that's a complicated mixture of myriad interactions and influences, then it behooves us to get as broad of sample of different types of brain measures and of different types of assessments as we can to see which one of these seem to be very influential on the other. And I'm certainly not going to walk you through all of these, but just to show you how many ways we can measure the brain these days, these are all measures that we get from MRI scans when we put our participants into the scanner. And so we're interested in how people's gray matter is aging, how people's white matter is aging, what neurotransmitter variables have to do with cognition and how the brain functions and how that changes over time. And then we can relate that to how well people do on these different cognitive tasks, like you saw on the scatter plots on the very first slide, and then how they are modified in some way by our health and lifestyle and genetic factors. And so the first thing I'll talk about is thinking about gray matter. And so the entire surface of your brain is a cortical ribbon of a gray matter sheet. And this gray matter sheet is composed of different cytoarchitectonic regions of the brain that differ in different properties. There's many different ways to think about these different properties, but the important thing for today is that we can measure the volume, the thickness, whatever metric we would like of these different regions of the brain. And when we've done this over the decades, we and other people who study this, we found that yes, the gray matter of the brain does thin and shrink with age, but it does so in quite a differential pattern. Meaning that some regions of the brain seem to shrink as just a part of normal healthy aging, and that seems to be okay. And other regions are preserved. And the regions that are preserved with normal healthy aging tend to be not necessarily preserved in some type of disease state. And we think that's the interesting predictive quality of doing these types of measures. So just as an example, and to set you up for some brain regions you're going to be hearing this whole hour, a lot of research is focused on the prefrontal cortex, both in normal aging, but also in disease, psychiatric and neurological diseases. And so if you take a region of the prefrontal cortex, as I've done here, you can see that based on the size of the brain of the 20-year-olds, by the time we are in our 80s and 90s, we've lost quite a bit of that brain tissue. This is cross-sectional results that I'm showing you right here. Longitudinal results look a little bit different, but you can see that this slope is quite steep. Matter of fact, this is the steepest slope in the brain here. And then another region, set of regions of the brain that you're going to be very interested for this talk is that regions of this more interior, more medial part of the temporal lobe. So here's the lateral portion of the temporal lobe. If you zoom on to the middle part, here's the middle part of the temporal lobe. Regions that sit adjacent to each other happen to have quite different aging trajectories, which we think is interesting and perhaps diagnostic. So here, for example, is the volume of the parahippocampal gyrus, and you can see that it has a significant decline with age. However, relative to the prefrontal cortex, it's not nearly as steep. And then its nearest neighbor here, the entorhinal cortex, which maybe you've heard of, and if you've heard of it, I'll bet it's probably in relation to Alzheimer's disease because it's one of the earlier regions involved in Alzheimer's disease. In a normal, healthy aging sample, you can see that this region of the brain stays pretty much intact across the lifespan. If I were to show you this same scatter plot in a group of individuals with, say, Alzheimer's disease, that slope would probably look more like this. And so one thing that we've learned is that regions of the brain can be not diagnostic, but they can give us a hint to maybe what type of trajectory an individual is on, whether it's more healthy or more pathological. And so these prefrontal cortex, these medial temporal lobes in the hippocampus and into the limbic system are very important to the study of aging, but you'll also hear and see that they're also of particular interest to psychosis. And so that's the gray matter. What about the white matter? Well, the white matter connections in our brain that connect to these proximal and distal gray matter regions, they also degrade with age. And so just like gray matter, we're finding that some white matter tracts degrade substantially with age, whereas others seem to be fairly well preserved. Here's a recent study that we've done where we honed in on some of these prefrontal limbic types of connections. And so we made these connections in the brain and we measured kind of how intact or how healthy these connections were in all of the different individuals in our sample, which are all these dots. And again, you can see these kind of look like the cognitive metrics that this property of the brain, that higher is better, it declines with age. And interestingly, this decline with age is significantly correlated with or predictive of how well people are performing on their tests of executive function. So the higher this number is when you're older, the better you're performing on these executive function tasks. And so these prefrontal limbic connections are especially vulnerable as we get older. They're significantly predictive of performance, and there's some of the exact sets of circuitry that are involved in psychosis. Okay, and so then now we can revisit the medial temporal lobe and the hippocampus. And so we've also shown that the hippocampus, as well as its inputs and its outputs, also change with normal aging, and these changes predict one's memory performance. And so here you can use a variety of different MRI techniques, like I showed you on one of those first slides, and you can look at the volume of different subfields inside the hippocampus. You can look at the major output of the hippocampus, which is this white matter tract here called the fornix. You can look at one of the major inputs to the hippocampus, which is the parahippocampal gyrus here, and you can look at its volume. And then if you put all these here together, you can see the circuitry fairly nicely. And so one of the things that we did is we looked to see how all these different regions of this important system change with age. And it appears that, unfortunately for us, all of these different regions of the brain that are involved in this important kind of memory circuitry do seem to be altered with age. Again, not on par with the level that it would be if this were some type of pathology, but this is just kind of what we can expect from our normal aging systems. And then probably most interestingly, the greater the age-related reduction, right? So there's all these individual differences where you are along this regression line predicts how good your memory scores were. And this specifically is on one of the types of memory tests that we all have problems with as we age, associative memory, like trying to put someone's name together with their face so you can remember them later. And this will be important again when you hear about our psychosis work in a couple minutes. And so I also mentioned neurotransmitter levels. This is our very own moderator here. This is work that he published in the lab. And we were interested in whether genetic polymorphisms surrounding how much neurotransmitter availability you have predisposition to, in this case, dopamine, how that relates to brain structure and cognition. And so here you can see are the major dopaminergic transmitter pathways in the brain. This blue one is the one of interest to us. And so we can go through and we can measure cortical thickness along the regions that make up this pathway. And then we can see, based upon your genotype on this dopaminergic polymorphism, you can see if individuals differ just based on this genetic variable on how thick these regions of the brain are. And in most of these regions, all but one here in this picture, we found that those individuals, these VAL genotype people, in every instance, they have lower dopamine availability, and that is shown to associate with the thinner cortex in these regions. And so these regions along this pathway, if you have this genetic predisposition to lower dopamine availability, you already have smaller brain tissue in those areas. And then you could say, well, maybe that doesn't matter. And so we asked if that mattered. And it turns out that that is a modifying mediating factor between results that we are aware of in the field, that if you have this genetic polymorphism, you tend to do more poorly on executive function tasks. And we've shown that one of the mechanisms for that, perhaps, is this smaller cortical thickness. And again, one of the reasons I chose these results to share with you is because this is one of the very, very important fundamental systems that's involved in psychosis and all types of psychiatric disorders. And so then lastly, what about brain function? Well, I left brain function for last for a reason. So brain function is most certainly altered with aging, but it's way more complicated. And so we understand brain structure, and smaller is generally poorer, less integrity in the white matter connections is generally bad. But with brain function, kind of the jury is still out, and we have a lot of work to do to understand this. And so on the one hand, some of the brain regions, when we put people in the MRI scanner and have them do complicated tasks, some of these regions increase their activity as we get older, and those are the warm colors here, and some of them decrease their activity as we get older in response to this cognitive challenge. And each of those predict poor cognitive performance. But if you look at the degree of connectivity of those regions during the task, we find that this level of connectivity does not change with age, at least in our studies so far. So this might be an avenue for something to think about that is more preserved with aging. It is important, though, because although it doesn't decline with age, as far as we can tell, the less you connect and correlate these regions with each other as you're doing these complicated tests, the less you're doing on the task in terms of accuracy, and the more poorly you're doing on some of the cognitive functions outside of the scanner. And so, like I said, it's more complicated and a very fruitful avenue for future research. There's even more complicated patterns when you look at this type of brain function measures, when you look at individuals with Alzheimer's disease or individuals with psychosis. And so the take-home message, I think, is that just like the individual heterogeneity, inter-individual heterogeneity I showed you across cognitive performance as we age, it's also there at least as heavily in individual differences in our brain structure and our brain function. And so there's individuals who have larger, for example, prefrontal cortex. There's individuals who have thicker cortex. There's individuals who are more and less susceptible to these kind of white matter lesions that we develop with age. There's differences in how intact these white matter connections remain as we age. And then again, kind of the jury's out on this one. There's definitely differences in how we activate our brains to different cognitive tasks as we age, and a lot of research is focused on that right now. So that's kind of the take home for me, is that there's lots of individual differences in all of these things as we age. And so I just wanted to thank the people that have worked on all of this with us, and all of course, the funding that we have to do this really expensive research. And thank you for your attention. I'm looking forward to answering your questions at the end of all these presentations.
Thank you, Dr. Kennedy. That was great. It was really interesting just to see how what we would classify as normal aging is so impactful to our brain structure and how that impacts our cognition. And especially it was nice to see all the different ways that we're going about studying aging with all those different modalities that you mentioned. So yeah, that was great. And I imagine our audience is also really curious about, okay, now we know more about what happens when we age in a typical way, but what about our brains, the brains of those who are not classified as normal? How do they age? And with my background in studying dopamine, I'm thinking about, okay, then what about those with schizophrenia? And I know you had mentioned that and psychosis. So is there anything different in that population? And I really think we get a nice answer to that question from our next presenter, Dr. Elena Ivelva, who I have the pleasure of introducing next. So Dr. Ivelva is an associate professor at the Department of Psychiatry, UT Southwestern Medical Center, and she also serves as the director of Early Psychosis Clinical and Research Program. In her research and clinical practice, Dr. Ivelva specializes on schizophrenia and related psychotic disorders. Dr. Ivleva's research is focused on understanding mechanisms of schizophrenia and developing brain-based biomarkers that can inform novel treatments. Dr. Ivelva approaches schizophrenia as a dynamic brain disorder evolving over the lifespan. And she also investigates the effects of aging and interactions between aging and illness itself. Dr. Ivelva's research has been supported by the National Institution of Mental Health, Brain and Behavior Research Foundation, American Psychiatric Association, and other foundation grants. Dr. Ivelva has presented her work nationally and internationally. She's an author of more than 70 publications and an editor of several books. She's a member of the American Psychiatric Association, Early Intervention in Mental Health Society, Schizophrenia International Research Society, and the Society for Neuroscience, among other professional organizations. So thank you for joining us today, Dr. Ivelva.
Thank you very much for such a nice introduction, Giuseppe. And thank you for having me as part of this event. Dr. Kennedy already started us off with a wonderful presentation on the research approaches she uses in her lab, as well as in broader field of normal aging. And what I will try to do here, I will try to build upon the information we just heard and move us to the realm of abnormal aging, such as how aging and illness course presents in psychotic disorders such as schizophrenia. And I'm also delighted to take this opportunity to talk about a very exciting collaborative project that we recently started between the Center for Vital Longevity at UT Dallas and Psychosis Research Program at UT Southwestern Medical Center. This collaboration allowed us to bring together two very fascinating, very complex, and I must say very different research fields. And I think that by combining our expertise, hopefully we will be able to contribute some meaningful outcomes and move both fields forward. So this is my presentation outline. I will start by talking about what is psychosis. Then I will share a bit of research from my own lab, and specifically we'll talk about how we approach schizophrenia as a dynamic, kind of evolving over lifespan brain disorder. Then I will talk about complexities of aging in schizophrenia. And finally, I will discuss our collaborative grant, Aging and Disease Course, Contributions to Lifespan Neurobiology of Schizophrenia. So first of all, what is psychosis? So in psychiatry, psychosis is defined broadly as gross impairment in reality testing or loss of ego boundaries. And let's just pause for a minute and think what this really means experientially for a person going through psychotic experience. And you know, when I was putting together this presentation, I thought, how would I do this in a talk like this? And perhaps to give us just a glimpse into psychotic experiences, I decided to show you some examples of art from a person with lived psychotic experiences. Louis Wain was an English artist who gained recognition as a painter and children's book illustrator. So he specifically liked animals and specifically liked painting animals. And one of the mostly portrayed animals in his work were cats. So Louis Wain lived with schizophrenia and spent the last 15 years of his life in a psychiatric institution. The psychiatric hospital was described as fairly nice, with a large garden and a very large colony of cats, to Wain's pleasure. And he continued to paint until the very last years of his life, even while he was confined to this psychiatric institution. So, I'm sorry, let me see. Yeah, so this is one of the examples of Wain's art from his earlier years of work. And you can see, you know, this is a cat, which looks like a cat. And if I look at it, it looks a little bit like a tapestry style, perhaps, so something like that. Now, over time, and with progression of his illness, his paintings began to change. So this cat looks a little bit different, perhaps, you know, can be described as a bit anxious or distressed cat. And then this cat has this kind of colorful halos emanating into universe, perhaps, as if the cat is dispersing into space. And then his later paintings began to look like this. And perhaps if we look at this last set of paintings, if we didn't know that this is a picture of a cat, it would be indeed very difficult to guess so. So I think these examples of art capture very well this definition of psychosis, and specifically this component, loss of ego boundaries. This is something that my patients with schizophrenia describe frequently to me, as psychosis is experienced as the state of highly fragmented reality, where different parts of reality do not quite join together, and where it is very difficult to tell what is real and what is not real. And one of the core complexities in psychotic experiences is that psychosis has exactly the same perceptual and broader cognitive qualities as what we call real experiences. So for the person going through this experience, it's extremely difficult to differentiate between the two. It typically is very confusing and distressing. If we go on to specific symptoms which define psychosis domain and disorders like schizophrenia or schizoaffective disorder, there are different kinds of symptoms here. So first of all, hallucinations, which are false perceptions in the absence of real stimuli. Hallucinations can span different sensory modalities. They can be, for example, auditory, when a person may hear noises or whispers, or perhaps conversations between people or entities which are not actually present. Delusions is the next type of symptom. Delusions are false beliefs held with conviction and are accompanied by loss of insight. Again, delusions can be of different types, different themes, but perhaps the most classic type of delusion is paranoid delusion, where a person may feel that they're being harassed or persecuted or plotted against by a person or group of people or some entities like, say, aliens or spirits. The next cluster of symptoms is thought disorder and cognitive dysfunction. And this is something I think is very important. So, when someone thinks about psychosis, typically people think about hallucinations and delusions and not necessarily about cognitive dysfunction. Well, it turns out that cognitive dysfunction is one of the very core symptoms of psychotic illness. And this is precisely what predicts how well the person is going to do on the long run. So psychotic symptoms themselves do not correlate with functional outcomes. However, cognitive dysfunction, this is what impairs and debilitates our patients, especially in later stages of the illness. Cognitive dysfunction is spanned over different domains, but the most difficulties are typically seen in different types of memory, such as working episodic memory and specifically associational memory component of episodic memory, difficulties learning new information, as well as slower information processing. And the final cluster here is impaired behavior, which is driven by underlying psychotic experiences. And while this behavior may be highly illogical to observer, it is in fact very logical to the person themselves. Sometimes it's referred to as psychotic logic. So if you think about it, if one feels that they're being, say, persecuted by a mafia or some other organization, it is in fact very logical to say stop answering calls or move to a different apartment or move out of town or to a different country. So this is very important to, I think, try to actually understand the experience of psychosis, how the person is going through. And recently we've published, we've completed this, very large comprehensive volume on psychotic disorders, which describes symptoms, neurobiology of psychosis, approaches to treatment. And I'm proud to say that on our cover, we used one of the Wayne's cats. So here I'm going to give you a few facts about psychosis. So first of all, it turns out that psychosis is fairly common, even though it depends how someone defines psychosis. About 1% of the population suffer from schizophrenia, and about 3.5% from psychotic disorders combined. So this would include schizophrenia, schizoaffective disorder, psychotic depression, psychotic bipolar disorder, and other conditions. At any given time, 2.5 million US citizens are affected by psychotic illness, and about 3 out of 100 will develop psychotic episode at some point in their life. If we broaden definition of psychosis from actual psychiatric disorders to what's called in psychiatry psychotic experiences, which are basically symptoms of psychosis, which do not reach the level of full diagnosis, it turns out that psychosis is even more common. So about 7 to 8% of the general population experience clinically significant symptoms, most frequently hallucinations. A simple and very common example, which many of us perhaps experienced, would be something like hearing your name being called or hearing your cell phone ring while it's not actually happening. But again, the majority of these individuals who have these mild psychotic experiences never develop full diagnosis of psychotic illness. And even milder psychotic experiences like unusual beliefs, illusions, and such are seen in up to 20 to 30% of general and generally healthy population. These experiences are most common in adolescence. And again, they resolve without any intervention, and the majority of these individuals never go on to developing full psychotic disorder. Schizophrenia and other psychotic disorders are usually diagnosed early in life. The peak onset is about 16 to 30 years of age. And because these are such important formative years of individual's life, the diagnosis and the illness itself have profound and long-lasting effect on the person. Longevity in psychosis is poor. So individuals with psychosis suffer from increased medical comorbidities. So they have more cardiovascular conditions, diabetes, autoimmune conditions. Individuals with psychosis die 30 years younger on average compared to people without the illness. And there is an alarmingly widening longevity gap between schizophrenia and general population, estimated to be about 37% gap increase from the early 1970s. About 70% of people with the illness experience suicidal thoughts at some point in their life, and about 10% die by suicide. Disability, unemployment, homelessness, and incarceration are unfortunately very common situations in this population. And the cost of schizophrenia from both direct and non-direct costs is about 156 billion per year. So how do we diagnose and treat schizophrenia and other conditions at present? So the diagnosis itself is purely clinical. And what I mean by this is that I, as a psychiatrist, gather information about patients' symptoms, behavior. I listen to the family. I may, have some observable behavior in my office, or somebody describes this to me. And this is how I make the diagnosis. So really, at the moment, we have no single brain-based test, despite the fact that this is clearly a brain disorder, which could help us to define our diagnosis of primary illness. Now, with that being said, we do run, brain MRIs and EEGs on our patients, but it is very important to understand that we do so not to establish our diagnosis of schizophrenia, say, but rather rule out other causes that may be associated with psychosis. For example, brain tumor or some sort of vascular abnormality. And schizophrenia in general, schizophrenia research field in general is a fairly mature field. So research has been done for a long time. And in the past about 20 to 30 years, the focus of schizophrenia research has been on neurobiological mechanisms. And we do have some bits and pieces of mechanism. However, at present, we still do not have the exact mechanisms of the illness. So in other words, we still do not understand why and how and at which points in individuals' lives exactly the brain starts producing, say, hallucinations or delusions. In the absence of mechanism, the treatments remain limited. So at the moment, the main treatment approaches are medications, and specifically antipsychotic medications, which suppress hallucinations and delusions fairly effectively. However, they do not have any positive impact on cognitive dysfunction. And in fact, at the present, there are no available treatments at all for cognitive dysfunction and kind of general decline in functioning. And we still have a substantial gap between clinical practice and research in our field. So from here, I'm going to move to the second part of my talk and tell you a little bit about my research and how we approach schizophrenia as a lifespan brain disorder. So first of all, schizophrenia, unfortunately, is typically a lifelong condition. Only about 5 to 7% of individuals who had initial psychotic break will never develop the second episode. So typically when somebody receives this diagnosis, it stays with the person for the rest of their life. From clinical perspective, we know very well that the symptoms of schizophrenia do not stay the same as the person progresses through the illness. So this diagram shows kind of a typical natural course of schizophrenia. And you can see that the first symptoms are typically seen early on, years prior to a person develops initial psychotic episode. And in childhood, these kids who are on path for developing schizophrenia may show some mild cognitive abnormalities, learning disabilities, psychomotor delays, social difficulties, and such. Then those individuals who in fact progress into further stages of illness move to the next phase, which is called Prodromal Phase, where we start seeing more of psychotic symptoms. And initially, they're usually kind of mild symptoms, very difficult to tell whether they're actually emergent psychosis or maybe these are still normal experiences. Then a proportion of individuals from here will move to the first psychotic episode, which is a full-blown, well-defined, clear psychotic state. And then in the early stages of illness, in the first approximately 2 to 5 years, psychotic symptoms tend to be very aggressive. So this is where patients require a lot of medications. They are frequently hospitalized. However, over time, psychosis tends to calm down, especially with treatment. But cognitive decline, social deficits, and general functional disability, unfortunately, tends to progress over time and gets worse as person ages. So we know this clinically and symptomatically, right? This is nothing new here. However, what we don't know, or know surprisingly little about, is that what's happening in the brain of these individuals in each of the stages of the illness. Furthermore, at the moment, we do not have any specific approaches to treatment for different stages of illness. In other words, we treat somebody who's been ill for, say, 6 months, exactly the same way as we treat a person with schizophrenia who's been ill for 30 years. And this is one of the kind of area of my research which I try to work on and move forward. So specifically, I'm interested to see what are the brain-based biomarkers or brain changes which may characterize early, intermediate, and advanced stages of schizophrenia. And our research already begins to show that, in fact, brain does not look the same across the stages. And if so, do the stages of schizophrenia require different treatments? So here I'm going to share you a bit of research data from my lab. So Dr. Kennedy already talked about the importance of hippocampus, prefrontal cortex, and associated limbic circuit in normal aging and in disorders of aging. The hippocampus and broader limbic system also happens to be the very hub of psychopathology in schizophrenia. So abnormalities in limbic system structure and function are known to contribute to cognitive dysfunction in schizophrenia, but we also think that they may be contributory to psychotic experiences as well. So here we did ultra high resolution brain imaging, specifically perfusion imaging. And because we used this very high resolution below 1 millimeter, we were able to look not only at the whole hippocampus, but also at different hippocampal subfields, which are shown here in different colors: dentate gyrus, cornu-ammonis 2, 3, 1, and subiculum. And we did this because these different hippocampal subfields are thought to be differentially involved in mechanisms of aging and schizophrenia. And here we had a sample of individuals with schizophrenia, chronic sample. So people were about 38, 39 years old on average, with illness duration of approximately 20 years, and age-matched healthy control. And when we initially looked at hippocampal activity, we did not really see anything interesting here. So you can see that, healthy controls, which are here in green, were very similar to schizophrenia group, which are shown in red. And this was true across all hippocampal subfields. So really nothing impressive. And believe me, the study was a very difficult, very labor-intensive study to do. And we were quite disappointed. But then we thought, well, before we give up on this, how about we do this? We decided to stratify our schizophrenia sample by illness duration. So we split these individuals into those who have been ill for not so long, less than 7 years, mid-course subsample 7 to 15 years, and advanced schizophrenia more than 15 years. And the pattern in the data looked very, very different. So what we found here was that individuals in early stages of schizophrenia showed this overwhelmingly high hippocampal activity across all subfields, while individuals who were later in the illness, mid-course and especially advanced schizophrenia, had the opposite pattern of hippocampal activity with reductions. And this is the same kind of data, same data actually, but plotted in a different way. So we did here a regression analysis, and we were able to derive a trajectory along the lifespan. So you can see how this hippocampal activity tends to change with progression of the illness. So it starts very high, then it seems like it drops pretty abruptly. So by about 5 or 7 years of illness, we see this massive decline in hippocampal activity. Then it tends to plateau for a little while, and then by approximately 25 or so years into illness, there is the second hit, and activity goes down from there. So in addition to people progressing through the illness, all individuals with schizophrenia, and all of us, for the matter of fact, inevitably age. there have been this pretty recent, actually, findings in our field of accelerated brain and cognitive aging. And Dr. Kennedy already talked about it a bit. So in healthy aging as well as in pathological aging, we are able to calculate one of these indices called brain age gap, which is the difference between chronological or actual age and age estimated from brain MRIs. And it turns out that we can actually estimate age of the brain from structural MRI at least pretty accurately. So this is really what constitutes brain age gap. And it turns out that in schizophrenia, there are these consistent findings of increased age gap, meaning that the brain of a person with schizophrenia looks older than it's supposed to look based on a structural MRI. However, the data are highly variable. Some of the studies show that there is a shift of approximately 2 to 3 years, while others show that it can be as high as 15 years. So this is the largest to date study which looked into that. So data from structural MRI from UK Brain Bank. So here they had thousands of individuals who were healthy controls, and again, thousands of people with different illnesses, including dementia, mild cognitive impairment, schizophrenia, bipolar disorder, major depressive disorder, and others. And what they did here, they looked at this brain age gap across different regions of the brain, which are shown here on the left. So frontal cortex, temporal cortex, etc. The warmer color indicate higher effect sizes or stronger effects, and the shift towards the right indicate older brain than expected. And you can see that dementia, perhaps as expected, looks the worst, but schizophrenia was just the second worst after dementia, even worse than mild cognitive impairment or, say, neurodegenerative brain disorders like multiple sclerosis. So really, this is pretty striking to me to see that, you know, our individuals with schizophrenia are not too far from, you know, individuals with neurodegenerative disorders such as dementia. However, you know, with this intriguing data in accelerated aging, there are a lot of uncertainties here. So one thing which is really not clear and not known is when in the lifespan this accelerated aging in schizophrenia happen. So some of the studies actually show that it may be a phenomenon that occurs very early in the illness. So one of the studies by Shang and colleagues showed that the brain age gap may be the highest in the first few years of the illness. So somewhere between the first 3 to 5 years or so, and then it tends to stabilize or kind of slow down. However, yet other group of studies show that this may not be the case. So for example, in this study by Zhang and colleagues, they looked at cortical thinning in one of the frontal regions, specifically ventromedial prefrontal cortex. And schizophrenia is here in orange and controls are in green. And you can see that the cortical thinning rates in schizophrenia were pretty remarkable, and they began really early in the illness. So by approximately age of 30-35, you see already the slope going down, and it goes down even more drastically after age approximately 50, which was not the case in healthy controls in this study. It is also not clear which of these phenomena, you know, aging versus illness scores, are explained by aging itself, whether this aging is the same kind of aging that we see in healthy people, and what can be explained by illness itself in the mix of this dynamic phenomena. And this brings us to our collaborative study. So once again, I would like to say that it's been, you know, a great pleasure and really a lot of fun and just an incredible opportunity to be able to do this collaborative project. So from the Center for Vital Longevity side, this work is led by Dr. Kennedy and Dr. Ragh, and Dr. Chastain is also involved in the study from Dr. Ragh's group. And at UT Southwestern, it is my lab, as well as Dr. Carol Taminger, who is my long-term mentor and the chair of the Department of Psychiatry, as well as several colleagues from the Advanced Imaging Research Center, Drs. Choi, Liu, Aslan, and Yuzhavash. And we were able to secure a 5-year grant from National Institution of Mental Health, which just began in July of this year. So again, I mentioned this, but once again, the central question here is which aspects of schizophrenia mechanisms can be explained by the illness itself, which by normal aging, and which by interactions between the two. So to support our hypothesis, we actually, developed this model, and I know I'm running a bit out of time, so I'll be brief here, just to say that we believe that we will be able to capture mechanisms specific to aging, specific to illness course, and we will be looking at interactions between these two different dimensions through multiple biomarkers associated with cognitive testing as well as clinical assessments. So very briefly about the study, what we are doing here. It's a large sample. We hope to recruit 168 individuals, half with schizophrenia and half healthy controls. We are recruiting over a broad age and illness duration span. Because we are studying this dynamic phenomena, it is a longitudinal study. We ask our participants to come in at initial time point and return after 2 years, and we will repeat all testing. And we will be targeting this aging and illness course phenomena through a very comprehensive battery of brain imaging with 4 different modalities, cognition testing, and clinical assessments. So our ultimate goal is to develop objective biomarkers and perhaps biosignatures for aging and illness course in schizophrenia, as well as for healthy controls, because I believe that from a study like this, we may have some important insights for normal aging as well. And if our model turns out to be correct, I think the study may have very important leads for future treatments and development of precision interventions. So indeed, if we find that, and if we confirm that early in the illness, hippocampus and limbic system and maybe broader brain systems tend to be hyperactive, then the treatment goal should be calming down this brain hyperactivity. And there are ways to do that. We can, for example, manipulate brain metabolites, such as inhibitory and excitatory metabolites like glutamate or GABA, or we can use inhibitory neuromodulation. Now, in advanced stages of illness, the treatment targets based on our model can be opposite. So this is where we need to actually get this system up and going. And here we may use different interventions, such as specific antipsychotics, which can increase brain activity, such as clozapine, or activating types of neuromodulation. And in parallel with that, there are very interesting novel approaches to promote healthy and successful longevity, including cognitive interventions, dietary modifications, metabolic control drugs, exercise programs, none of which are currently used in individuals with psychotic disorders. So we hope that by studying aging and drawing attention to aging processes in this vulnerable population, we will be perhaps able to develop sub-targets for successful aging as well. And with that, I would like to thank all my colleagues at UTD and UT Southwestern, all the funding agencies that have been supporting this work over the years, and thank you very much for your attention and time.
Thank you, Dr. Ivelva. That was great, and it was really interesting to see how schizophrenia impacts individuals across their whole lifespan, which makes sense why we see significant differences in overall aging processes between those diagnosed with schizophrenia and those classified as being in the normal population. And also, I thought it was really interesting to see the differences in illness durations for those with schizophrenia. And also, when you mentioned towards the end, the accelerated aging aspect, I thought in particular was interesting. And it's going to be great just to learn more about the aspects of those mechanisms of schizophrenia and how they're explained by both aging and the illness itself, and also those interactions that you had mentioned. So yeah, thank you so much to Dr. Ivelva and Dr. Kennedy for your presentations. We're a little short on time today, but we have time for a few questions from the audience. And I think we do have one for Dr. Kennedy. Okay, so the question is, what is going on in older adults who age very well? I know their brains look younger usually, but is there any idea to why they're aging so well?
Yeah, that's a good question. It's the question, honestly. And so part of this is how science develops. And so in the beginning, most research was focused, and as it should have been, on pathology. So let's learn as much as we can about Alzheimer's disease and other neurodegenerative diseases so that we can treat it. And then once it became apparent that to understand pathology, you kind of have to understand, well, what's the other edge? Where does normal lead up to before it hits the cusp? And so that body of research, my field, is younger. And so in the beginning of that field, it was almost felt a little recursive and inverse, like I said in my talk, that, well, your brain looks really good and you're aging well because your gray matter isn't shrinking, your white matter connections aren't deteriorating, et cetera, et cetera. And so now, more recently, and this is all as our tools as scientists evolve and get better too, we're able to ask more mechanistic questions now. And that is why I think all of these aging studies are now finally needing to turn to studying individual differences. And so you can think of aging like anything else, right? This whole nature versus nurture. And unfortunately, maybe, you know, nature takes a little more control in something like psychosis, but in aging, it seems to have less of a role, thankfully, right? And so if you think about the nature versus nurture, you have a whole lifetime to live before you're old. And so all of the things that you put kind of in the good column that add up, some of them genetic, but the genetic factors for aging seem to be very small, single-digit percentages of variance in any type of brain metric or cognitive metric that you look at. So the inverse of that is that it's good. You know, you kind of get dealt this set of cards when you're born, and they have a little bit of influence, but it's really how you live your life. So all of the things that you know that are good for you, right? Exercise and proper diet and all of these things accumulate across the lifespan to help you age. And so I think the people who are aging the best kind of got the best start genetically, got the best start socioeconomically and educationally. And then once you're an adult, it's just kind of keeping up these good habits. And so as we get better tools to look into more of like the mechanisms of why the brain is staying healthy, we'll start to have a better answer to this. But that your question is the goal of my whole field, and we're just not there yet.
Okay, thanks for answering that. And I think the next question, well, you had spoke a little bit about genetics, Dr. Kennedy, but this question I think is for Dr. Ivelva. The question is there evidence individuals with schizophrenia have genetic abnormalities in regards to dopamine?
Yes, this is a very good question. You know, genetic field is one of the kind of older fields in schizophrenia in a sense. And initially the hope was that we could identify the gene or maybe 2 or 3 genes which could explain the illness. So it absolutely failed. And what we know by now that this is absolutely incorrect. So this is definitely not a disorder that is driven by 1 or 2 or 10 genes. So where the data stand in the field, so this is the data from the largest schizophrenia genetics consortium, which looked at over 150,000 cases with schizophrenia and did GWAS, so whole genome studies, trying to identify those genes. And they were able to identify 108 loci in the whole genome, which seem to contribute to schizophrenia. So basically those 108 loci cross the level of statistical significance at the level of whole genome. Now, each effect at each of those loci was very small. And what seems to be also important is the interactions between different loci. So, you know, it is not just that, you know, so really there seems to be no strongly strongly defining genes, but it's the combination and the mosaic, this very diverse genetic interactions which seem to contribute to illness. Now, perhaps I can also answer it from a little bit different end. So if you look, you know, at heritability of schizophrenia, right? So the rates of schizophrenia in a first-degree relative of somebody who has schizophrenia, so that would be, you know, mom and dad, or first-degree, you know, a full sister or brother, or biological child, is approximately 8 to 12%. So this is the risk. So in general population, it's 1%, and in first degree relative is about 8 to 12%. So it is a substantial increase, right? 12 times increase. However, it is not 80%. So this once again, brings up the topic of, yes, genetics contributes, but there are many other environmental, just kind of innate biological factors, epigenetic factors as well, which play into this illness.
Okay, great. Thank you for answering that. We do have one last question, and it's an interesting question. It's what is the justification for including patients with both schizoaffective and schizophrenia disorder instead of just schizophrenia, and if we do not know the exact mechanisms underlying those disorders?
Yeah, no, this is a very good question. So, clinically, we know what's the difference between schizophrenia and schizoaffective disorder. Just very briefly, in schizophrenia, it's the psychosis as well as cognitive impairment, which are primary. In schizoaffective disorder, we add an affective component, and that's the schizoaffective part, which can manifest as either depressive or manic or hypomanic episodes. So that's the difference. So really, people with schizoaffective disorder, they kind of get the full load of both psychosis and mood or affective abnormalities. Now, there have been quite a few studies, including our own work in structural imaging and other type of imaging and cognition as well, which show that biologically, these two conditions are not very different. So if you look at the brain of individuals with schizoaffective disorder and schizophrenia, they in fact, in our work, they don't separate statistically, say from the structural MRI perspective. So, since this study is definitely, study emphasizing neurobiology and looking at brain mechanisms and, potential biomarkers, we felt that it is appropriate to combine these two diagnostic groups, because again, biologically, I do not believe that they're very separate.
Okay, perfect. Thank you for that insight. That's really helpful. And thank you, everybody, for all the questions. And thank you again, Dr. Kennedy and Dr. Ivelva, for your presentations. And I'd like to hand it back now to Enrica to wrap up today's talk.
Thank you so much for sharing your time, your knowledge, and your resources with us today, Dr. Kennedy, Dr. Ivelva, and Giuseppe. And we also want to say that today's discussion was held in recognition of Alzheimer's Disease Awareness Month. For more information, you can visit the UTD School of Behavioral and Brain Sciences online, or click over to the Center for Vital Longevity to read more about the amazing and important research that is going on in both locations. And audience, thank you for joining us today, and we hope to see you all again in 2022 for more Comet Corner sessions. To view a full list of the Office of Research and Innovation events, including all that are listed on the Comet calendar at UTD, please visit utd.link/research calendar or scan the QR codes on your screen. Thank you and have a nice afternoon, everyone.